right. Great. So we have a pretty full virtual room here today, which is excellent. Great to see. And I'll kick things off. So I am very pleased to welcome Dr. Armin Raznahan to our genetics colloquium today, where he's visiting us virtually from the National Institute of Mental Health, where he is a child and adolescent psychiatrist and an intramural senior investigator. Armin got his undergraduate and graduate degrees in the UK, including in medicine and pediatrics at King's College University in London, and psychiatry at the Maudsley Hospital, also in London. And then he moved over to the NIMH, where he did his postdoc training with Dr. Jay Geed and Dr. Judith Rappaport, where he led a number of impactful neuroimaging studies that were broadly focused on neuroanatomical changes in the human brain across development. In 2015, he joined the NIH Lasker Clinical Research Scholars Program, and as of 2020 is now a tenured senior investigator at the NIMH Intramural Research Program, and he is the chief of the section on developmental neurogenomics. Armin's lab uses neuroimaging and transcriptomics in human participants and model systems to study brain development, with a frequent focus on sex differences. His lab has also published innovative work taking a close look at sex chromosome aneuploidies in human patients and models as a way to better understand how sex chromosomes contribute to brain structure and function. I first met Armin years ago when he spent, was it a few months, in Dan Geschwin's lab at UCLA, where he was learning the ropes of transcriptomics and bioinformatics analyses at the time. And I was a grad student in Dan's lab then, and Armin and I shared this interest in trying to understand the contributions of sex differential biological factors to brain development and developmental disorders. And we've crossed paths a few times since then. And I've always been intrigued in how Armin's group is able to intersect neuroimaging from living human patients with genomics data, and how his work on patients and models with sex chromosome aneuploidies can really inform our understanding of the role of sex chromosomes in neurobiology. So all that said, I am glad that Dr. Raznahan is able to join us today and share some of his group's latest work. And welcome, Armin.
Thank you, Donna. Thank you so much for the kind introduction, for the invitation to come and have the chance to speak with you all today. Can you all see the main expanded screen?
Mm-hmm.
Great.
Yes.
Yeah, it's wonderful to be here. You took me back to some happy days in UCLA with the palm trees and fine beaches, which we don't have too much of in DC. But and it was lovely to get to know you then and to be in touch since. And it's been a great day of meetings with you and with others there. And I hope it's the beginning of more conversations to come. So as Donna mentioned, what I'd like to do today is to talk a little bit about some of the work we've been doing, thinking about sex chromosome dosage effects in human health and disease. And an important question to ask, I guess, right off the bat is why pluck out these two for special treatment when you've got a whole bunch of other genome that you could be looking at. The reason we're particularly interested in sex chromosomes is because we think of them as sitting at the intersection of three areas of particular interest for our lab, which, as Donna mentioned, is a clinical research laboratory broadly interested in understanding the biological bases of risk for neurodevelopmental disorders. So sex chromosomes are foundational to the biology of sex and are a strong candidate biological contribution amongst many other contributions to any phenotypic differences one might observe between XX females and XY males. And that's a particular interest for a child psychiatrist because sex is a major modifier of risk for psychiatric illnesses in early development. A second lens of interest that draws us to sex chromosomes is from the viewpoint of psychiatric genetics. So we know that individuals carrying abnormal numbers of sex chromosomes, I'm showing you here in the columns, females with an extra X, males with an extra X, males with an extra Y, show rates of psychiatric illness that are substantially elevated compared to those seen in karyotypically normal, average XX females and XY males. And so this tells us something about the capacity of sex chromosome dosage to modulate risk for psychiatric illness. and therefore they provide very potent models of genetic risk for cytopathology more generally. And the third lens of interest that brings us to focus on sex chromosomes is that they're fascinating models for broader evolutionary and regulatory aspects of genome biology. So through the lens of understanding sex chromosome dosage effects, we can think about how they provide a model for how the genome handles gene dosage variation, particularly through the example of X chromosome inactivation, which I'm showing here in this cartoon, the way in which all female cells with two X chromosomes, or one of the X chromosomes, will be randomly inactivated and condensed into this peripherally located bar body, the inactive X chromosome. So by studying sex chromosome dosage effects, we get this ability to understand how the genome does this exist-mediated silencing of one of the X chromosomes in each female cell. And also the fact that the sex chromosomes have undergone these separate sort of evolutionary histories by virtue of their sex-linked nature means that sex chromosomes provide a lens into what happens when sequence homologous genes stop recombining and become exposed to distinct selective pressures in the same species. So before we dive into the meat of the talk, I just wanted to spend a few slides unpacking these unique features of sex chromosomes, specifically unpacking the unique organizational features of the sex chromosomes, which kind of give rise to them being interesting to study through the three domains that I've just mentioned. So just to quickly overview the evolution and anatomy of sex chromosomes, the current model is that about sort of the 300 million years ago, the chromosomes that were to become the modern X and Y chromosome were just like any other pair of autosomes, fully sequence homologous and recombining along their length. And then the current model is that about 300 million years ago, the chromosome that was to become the Y, either through mutation or transfer from another place in the genome, obtained a major sex-determining gene that essentially encoded for male sex. And that further along on that to become Y chromosome was an allele that was sexually antagonistic, so promoting a fitness in one sex and not in the other. So the presence of these two then favored the retention of translocations, of inversions that prevented the recombination separating these two loci from each other. So that when you had then an inversion of the chromosome that was to become the Y, this whole slab was then prevented from recombining with the X as it had been able to. And the current model is that what this led to over time was a series of sort of segmental separations of the Y from the X due to inversions on the Y. And then you have these genes on the Y that are no longer kind of kept clean and healthy through recombination. They only ever exist in males and they're not recombining with anything. And that promoted gene loss. So leading to a current situation where you have a very small Y chromosome with few genes and compared to the size of the X chromosome, that the Y essentially has lost over time almost all of the genes that it used to share with the X. And that leads to a very interesting situation where I'm showing you this cartoon here of the X and Y chromosome, and I'm using different colors to identify different families of genes, which differ from each other in the way in which they're unique to the X or the Y or shared between the two. So and I'm showing you this because it's a sort of important bedrock for the data slides I'm about to get into for the rest of the talk. So the X and Y still share in common these blue regions at their tips, the pseudo-autosomal regions, which are sequence homologous and continue to recombine. They're the remnants of the homology that was there when they were autosomes. And this homology and this recombination at these points is important for correct segregation of the sex chromosomes in cell division. The only other family of genes that have some similarity between the modern X and Y are shown here in purple. And these are a fascinating family of genes that you'll see become very interesting later on in the talk. They're called gametologs, XY gametologs. In some ways, these are sort of survivors of the wreck of the Titanic, they've been called by David Page, who's a prominent researcher in the field. These are the ancestral genes that the X and Y used to have in common that for some reason have still been retained on the Y. They've not been lost. So even though they're no longer recombining with their previous, what used to be partners on the X, they've not been lost from the Y, hinting that they may have particularly important biological roles. You'll see that I've colored these in slightly different shades of purple because they have very high sequence homology, these pairs. For example, ZFX on the X and ZFY on the Y. The sequence homology is typically above 90%. But it's not clear that these genes are actually still functionally homologous. And there's reason to believe that in some instances, the regulation of the X and the Y member has started to diverge. Other than these two families, which have some homology on the X and the Y, one recombining, the pseudoautosomal, and the others not, the gametologs, you have the remainder of the X, which is X-linked and only exists on the X, and the remainder of the Y, which is Y-linked and only exists on the Y. So as already mentioned, that leads to some very interesting consequences for gene dosage in XX females and XY males. Both sexes have 2 copies of pseudoautosomal regions. The X-linked gametologs exist in 2 copies in females and 1 copy in males. The Y-linked gametologs only exist in males. The X-linked genes exist in 2 copies in females, and the Y-linked genes exist in 1 copy in males. And what I'm showing you down here is the process of X inactivation through which one of the 2 X chromosomes in females is silenced. So here you have a DAPI stain showing 2 nuclei in an XY cell line and nuclei in an XX cell line. The X stain identifies the single X chromosome in males and the 2 X chromosomes in females, and the Y stain uniquely identifies the Y chromosome in males. And of these 2 X chromosomes in females, here you have female cell lines with the 2 X's identified, but also with a layer of RNA fish for exist, which is the molecule that initiates the process of inactivation of one of the X's. And you can see how this exist locus co-localizes with this peripherally located X chromosome territory, this compressed inactivated X chromosome. So what that means is that of these 2 X chromosomes in females, these 2 copies of the X-linked genes, the 2 copies of the X-linked gametologs, one of these are thought to be mostly, although not entirely, silenced on one of the X chromosomes. So this interesting patterned disparity in different families of sex chromosome genes means that right from the get-go, before you think about the sex differences in circulating gonadal steroids or the profound sex differences in the experienced environment, this sets up a very foundational sex difference between male and female cells in terms of the dosage of genes, which could potentially contribute, as I said earlier, to phenotypic sex differences. But sex chromosome dosage variation in humans is even richer than that. And what I'm showing you here are examples from a family of disorders that we study intensely called sex chromosome aneuploidies. And these occur when individuals have sex chromosome dosages other than the typical XX for females and XY for males. And you can see there's this very wide range of dosages. So in females, you can have individuals with 1X, 2X, 3Xs, 4Xs. In males, you can have karyotypically normal males, XY, males with an extra X, males with an extra Y, males with an extra X and an extra Y, males with 2 extra Xs. And collectively, this family of conditions occur in about 1 in 400 births. And the most studied of them from a medical perspective are Turner syndrome, most commonly caused by 1 X chromosome in females, and Klinefelter syndrome caused by carriage of an extra X chromosome in males. And these conditions are fascinating to study for some of the reasons I've already alluded to. One of these I just want to highlight, as is sort of conveyed by this table. You have, as a family together, they represent these very powerful sort of dissociations, differential copy changes of different family members. So if you want to do genotype, phenotype mapping studies, you can take families of sex chromosome aneuploidy cohorts, characterize how a given phenotype, let's say brain size or a behavioral variable or any phenotype of interest, varies across these sex chromosome dosage groups, and ask whether that variation for a given phenotype best tracks any one of these gene families that are differentially modified in their dosage. So one of the strong motivations for studying sex chromosome aneuploidies isn't just the lens they give us into sex chromosome dosage effects. So for example, there we're thinking of these medical disorders as model systems in humans that help us kind of pass sex chromosome biology, elements of sex chromosome biology that may be operative in health. But we're also interested in studying them as a lab within the NIMH because of their clinical effects on human brain structure and function. So if we think about behavior, for example, let me orient you to this plot. Each of these points on the x-axis, it captures a continuous measure of behavior that can be altered in psychiatric populations. So people can vary in how depressed they are, for example, or the level of somatic problems they have, or their social difficulties, or their attentional difficulties, or their tendency to show rule-breaking difficulties or rule-breaking behaviors. And the value of these measures on the scale that we use here in the general population is here at 50. And the x-axis is showing you the t-score of these measures in different sex chromosome aneuploidy populations. The higher the dot, the more severe the impairment of the aneuploidy group in that particular domain of psychopathology. So what this is telling you is across multiple different facets of behavior, there's increased risk for problematic symptomatology, for mental health symptomatology across all of the 4 aneuploidy groups shown here. Females with an extra X in green, males with an extra X in orange, males with an extra Y in pink, and males with an extra X and an extra Y in blue. The other important take home is that, a second important take home is that within any given aneuploidy group, it's often attentional problems and social problems that are amongst the highest. So it seems that sex chromosome aneuploidies may have somehow a particularly strong impact on domains of social and attentional functioning. And the third important take home is that there seems to be a separation between the symptom profile of males carrying an extra Y as compared to groups carrying an extra X, suggesting that which chromosome you carry might impart a different profile of risk, with the X carrying potentially more risk than the Y for some of these domains. And these patterned effects of sex chromosome dosage on human behavior are accompanied by patterned effects of sex chromosome dosage on brain anatomy. What I'm showing you here is a spinning view of the cerebral cortex and a spinning view of a zoom in of subcortical structures, the striatum and the thalamus there in the middle. Where you see a color, that region statistically significantly changes its size if you carry an abnormal number of sex chromosomes. And where you don't see a color, that region seems to be anatomically insensitive to sex chromosome dosage. The color encodes which chromosome is having the effect. So these red regions significantly change their size whether you carry an X or an extra X or an extra Y. The green regions seem to preferentially change their size if you carry an extra X, and the blue regions preferentially if you carry an extra Y. So I hope this already sort of starts to hint at some of the power of using these sex chromosome aneuploidy models. You can get these double dissociations with different phenotypes tracking different chromosomes. So with that context, I wanted to spend the rest of the talk focusing on a series of recent studies from our group that have tried to address parts of this multi-level model that sketches out the steps that we imagine are likely to be relevant between how many sex chromosomes you carry and brain structure and function. So presumably, the effects of sex chromosome dosage on the brain are most proximally reflected in the effects of sex chromosome dosage on gene expression, which then presumably influences cellular structure and function, which over development might shape circuit formation at the microscopic level, and potentially also the organization of brain systems at a larger scale of resolution, how the front of the brain might be interacting with the back, for example, structurally and functionally. And we've been trying, perhaps foolishly, given the distance, the biological distance traveled by this cartoon, to understand different steps of the process and to try to link together a kind of integrated model that follows the breadcrumb trail between sex chromosome dosage changes and alterations across these various levels. And that's what I wanted to talk through a little bit now. So I'll first begin with some early studies. These date back to the time that Donna was referring to when I was first learning transcriptomic analysis with Dan Geschwin's group. And this is a study where we try to systematically map sex chromosome dosage effects on gene expression in humans using then microarray measures in lymphoblastoid cell lines. Then I want to tell you about a newer series of studies where we've updated this approach by expanding the number of participants we're looking at using RNA-seq and asking, firstly, can we replicate these earlier published effects of sex chromosome dosage on gene expression in lymphoblastoid cell lines? But then can we also ask to what extent are the effects of gene dosage stable across different cell types? So we're comparing sex chromosome dosage effects on gene expression in lymphoblastoid cell lines, fibroblasts, and iPSC-derived neurons. And then finally, I want to share a study that tries to jump scales of analysis, where we say, is there any way in which knowing something about sex chromosome dosage effects on gene expression at the cellular level is in any way related to the spatial patterning of sex chromosome dosage effects on regional brain anatomy? So let's start with the microarray studies first. And this was our first serious look at sex chromosome dosage effects on gene expression. So we had 7 sex chromosome dosage groups that I'm showing here, and we had about 10 individuals per group, and we had microarray measures of expression of about 20,000 genes, and this was done in close collaboration with Dan Geschwin. What I'm showing you here is the pairwise comparisons between individual groups out of the 7 in our study design, and how different pairwise comparisons capture different dosage alterations. So for example, when you compare 1x to 2x, that contrast is capturing a difference in X chromosome dose. When you compare XY with 1x, it's capturing a difference in Y chromosome dose, etc. And in this study design, we then have multiple pairwise comparisons that capture an X dose effect and multiple pairwise comparisons that capture a Y dose effect. So the first thing we did is screen all 20,000 genes to say, are there any genes within the system that always statistically significantly change their expression if you mess with X chromosome dose, or always change their expression when you mess with Y chromosome dose? And of all 20,000 genes, 10 genes met this criteria, which we refer to as obligate dosage sensitivity. These genes included exist, reassuringly and unsurprisingly. That's a gene that's responsible for shutting down any X chromosome other than 1. And you can see that exist expression is high in the groups that have 2 X chromosomes. It included 4 a set of genes, 6 genes that reside on the X chromosome, which had been annotated as escaping X inactivation. So you can see their expression is elevated in groups that, I apologize, only 3 others, elevated in groups that carry 2 X's and lower in groups with 1 X. And then it included a set of Y-linked genes, which are higher in groups with excess Y chromosomes. And interestingly, this family of sex chromosome genes that show obligate dosage sensitivity was enriched for these gametolog genes. You remember, these are the sort of evolutionarily preserved sex chromosome genes that I mentioned earlier that seem to have somehow been retained on the Y chromosome over millions of years of evolution, when the rest of the ancestral genes on the Y were lost. So this was helpful in finding a kind of family of highly dosage sensitive genes on the sex chromosomes. The next question we asked using this model was, could we recapitulate the theoretical prediction for how different families of sex chromosome genes would change their expression as a function of sex chromosome dosage? So forgive me, the color key has changed here, but you remember I told you earlier that you can split up sex chromosome genes into the pseudo-autosomal regions that exist on both an X and a Y, Y-linked genes that only exist on the Y, and then X-linked genes. You could divide them into two families, those that are annotated to escape X inactivation, so they're expressed from each X chromosome in the cell, versus those that are annotated as undergoing full X inactivation, and therefore the idealized theory would predict that they're only expressed from one X in every cell, no matter how many X's you have. So the a priori model that existed at the time we did this study for sex chromosome dosage compensation was that you would have this number of copies expressed of PAR genes, which is essentially how many sex chromosomes the group has. This number of Y genes expressed, which tracks with how many Y chromosomes the patient group has. This number of X chromosome escape genes expressed, which tracks with how many X chromosomes the group has. And then you would predict that only one of the inactivated genes is expressed in any of these groups. So that sets up a four-class model prediction. And we used a fully data-driven method to cluster sex chromosome genes based on the observed profile of their expression across these 7 groups, and asked, do we recapitulate the theoretical prediction? And I'm showing you the results here in these plots. Let me just orient you to these. On the X chromosome, you have the 7 sex chromosome dosage groups, or on the X-axis rather. On the Y-axis, you have the mean expression, and each square relates to a different empirically defined cluster of genes, in this case, a single gene. So for example, let's look here at exist. You can see the expression of exist ramps up as the number of X chromosomes goes up. It is low in groups with 1 X chromosome, high in groups with 2, and higher still in this group with 3 X chromosomes. The PAR cluster, I'm showing you the theoretically predicted profile of expression in this dotted line. The PAR cluster, the observed colored expression perfectly overlapped with the prediction. So the expression nicely followed how many sex chromosomes you have. We also detected a Y gene cluster, which almost perfectly, although not entirely, followed how many Y chromosomes you have. And then we discovered 2 families of X chromosome genes. One, which was enriched for genes that undergo X inactivation, which showed a pretty flat expression profile as predicted. And another one, which was enriched for genes that have been annotated to escape. And although this showed some dynamic changes as a function of how many X chromosomes you have, they didn't quite match the prediction. They were more muted than the predicted expression profile. And what I'm doing here is I'm zooming in on these 2 squares a bit more closely. And I just want to draw your attention to a few properties of this plot. Individual genes in this green and yellow cluster are shown here in this plot for reference. Is that you're seeing this family of escape genes showing less dynamic expression than you'd expect. So something is further dampening their expression. And this family of X genes that are subject to X inactivation, although they look flat at this scale, when you zoom in, you actually see some differences in their expression as a function of sex chromosome dose. And although these are small in absolute magnitude, these differences were statistically significant in the data set that we examined, and they've been replicated since. And this is really intriguing because what this told us is that there's a family of X chromosome genes that are actually more expressed in groups with just a single X chromosome than they are in groups with multiple X chromosomes. And if there's time, we can talk more about this in discussion. But what we think this is pointing towards is that having an inactive X in the nucleus is somehow having a trans repressive effect on the active X chromosome. And when you remove that inactivated X, there's a break that's taken off the other X, the remaining X, and the expression is actually increased. So the importance of this work is that it's an example of how what we found interesting about this work is that it uses clinical populations to test a model regarding a normal process of dosage compensation of sex chromosome genes. And we broadly found support for the model, but we found interesting departures from the model. And what these departures mean is that they define new sets of genes that we now believe to actually be unexpectedly differentially expressed between typically developing males and females, even though the observed count of the genes might suggest that they wouldn't be differentially expressed, or to show differences in expression that are less marked than one might have predicted. So this was using microarray in a relatively small sample in lymphoblastoid cell lines. We've since been able to push this a little bit harder in an updated study design. And this is work led by Siwan Liu within the group. And I just want to orient you to these slides. So it's a similar strategy. We have groups here, 6 groups that differ in their sex chromosome dosage. And we have 3 cell types now: lymphoblastoid cell lines, fibroblasts, and iPSC-induced neurons. You'll notice that whereas in the microarray study we had 10 individuals in each group, for lymphoblastoid cell lines we have many more individuals in each group. We have a smaller number represented in fibroblasts and smaller still in iPSC-induced neurons. There were only 2 in each group, and the objective here was really to look at trend effects. But what I want to focus on initially is whether we were able to replicate our microarray studies in this new larger lymphoblastoid cell line set of biosamples using RNA-seq now. And what Siwan has done here is he's shown this pairwise comparison between all the groups we had, again, using shading to encode the dosage disparity that each contrast captures. So for example, here, if you compare XXYY with XYY, they're disparate in how many X chromosomes they have, and they're disparate in the total number of sex chromosomes they have, so in their par gene dosage. Whereas if you look at this contrast, XYY versus XX, this contrast is disparate in how many sex chromosomes, how many X's, and how many Y's. So, and there are multiple such contrasts that differ in what they capture between the 6 groups we have. So, in the first analysis we did, we searched for these genes. Are there any genes that show obligate dosage sensitivity? Are there genes that always change their expression when groups differ in X dose, always change their expression when groups differ in Y, or always change their expression when groups differ in the total number of sex chromosomes? And we found that indeed that was the case. And the RNA-seq data largely reproduced and extended the microarray data. So now we have 18 genes that show obligate dosage sensitivity to X chromosome dose. They include ones that we found in the microarray study, but they extend this list. And again, they overrepresent these gametologs. These 18 genes on the X out of the several hundred the X carries are enriched for genes on the X chromosomes that have somehow retained a copy on the Y. Again, on the Y chromosome, we reproduced many of the genes that show obligate dosage sensitivity from our microarray analysis, but now extend the list. And again, the family of Y chromosome genes that seem to consistently, in an obligate way, change their expression when you change Y chromosome dose are enriched for these gametologs. And as expected, we found that if you say which genes always change expression with sex chromosome dose, no matter if it's an X and a Y, you recover genes on the pseudo-autosomal regions, because they're the regions of the sex chromosomes that always change their dose as a function of how many sex chromosomes you have. So we think this is important because if you're wanting to ask the question, because the sex chromosomes are, the Y is small, but both the X and the Y have plenty of genes on them. And if you're interested in saying, well, how can it be that carrying too many X's or carrying too many Y's puts you at increased risk for altered brain organization or mental health issues? What one would really like is some way of prioritizing, narrowing the search. Are there particular genes that the data suggest we should focus on first as genes that could be driving the downstream effects of sex chromosome dosage? And this criterion of obligate dosage sensitivity, we believe this is the family of genes that we'd like to focus on first, because it seems that they're always shifting their expression, and therefore they're strong candidates for driving downstream effects. And this is just a quick slide. For reasons of time, I'll run through it, but it essentially shows that we reproduce this four-class model using RNA-seq data in the new study design. So again, fractionating sex chromosome genes based on their observed expression response to sex chromosome dosage empirically recovers these theoretical sets of genes that we would expect to behave differently in their expression. But everything I've told you so far is in lymphoblastoid cell lines, which are immortalized peripheral white cells. So that, frankly, they don't even exist in nature. They're a sort of idealized cell type. And as a neuropsychiatrist, I'm particularly interested in the brain, in brain cell types. But more fundamentally, the question that then prompts is, how reproducible are these effects that I've just told you across cell types? And there are a couple of reasons that I think that's a really important question. One is from a genome biology perspective. To what extent is it the case that do different cells show a stereotyped response to a dosage alteration of the same set of genes? If they don't, what factors determine why one cell type is responding in a particular way to a dosage alteration of gene set A, whereas another cell type is responding differently? Where do those differences come from? And that question, I think, sheds interesting light on genome regulation. But from a more clinical and pragmatic perspective, this question of how reproducible are transcriptional effects across cell types is important if we ever want to use these transcriptional data, for example, as a candidate biomarker or a clinical index in patients, because we aren't going to be able to convince large numbers of patients to give us their brain tissue while alive. So what one can, however, do is draw peripheral tissue from patients. So if nature throws us a bone, if there are certain aspects of the transcriptional response to, say, sex chromosome aneuploidy that are stable, that are consistent between a peripherally accessible tissue in the brain, then maybe we can zero in on those, and they might provide us a way of measuring, for example, inter-individual differences in dosage compensation between different patients carrying the same aneuploidy. And those differences could then, that we measure peripherally, could then help us potentially predict what's happening centrally. So to get at this question, what C1 did is he rooted the analysis in the large data set of lymphoblastoid cell lines. I'm just going to orient you to this plot here. So what C1 wanted to do was to say, are the genes that show differential expression in any contrast? So this column, it refers to the genes that are differentially expressed when you compare trisomy X with Klinefelters, for example. He first defined the genes that are differentially expressed in each of these contrasts in lymphoblastoid cell lines. And then he said, are these genes also differentially expressed in fibroblasts? And he asked that question, stratifying the genes, the differentially expressed genes, by whether they resided on the X, on the chromosome that's altered in dosage in each of these contrasts, or whether they reside on the autosomes. And what these red boxes are telling you is that there was significant agreement in the differentially expressed X-linked genes in lymphoblastoid cell lines and the differentially expressed X-linked genes in fibroblast cell lines when you mess with X chromosome count. But there was not preservation of the differential expression seen in autosomes. So what this plot is telling you is when you consider alterations of the X chromosome and you compare lymphoblastoid cell lines to fibroblast cell lines, there is cross-tissue consistency in the differential expression of X-linked genes, which is sort of one might predict given that they are the genes that are altered in dosage, but not cross-tissue consistency on autosomal genes. And this motif, a red bar up here, but more gray down below, is also what C1 saw when he asked the same question, now comparing LCLs with induced neurons. And he asked the same question again, this time focusing on Y-linked genes. And again, when you alter Y chromosome count, the stability between tissue types in differential expression is high for Y-linked genes, but low for autosomal genes. And that broadly is similarly seen when you compare LCLs and induced neurons, with some interesting exceptions. So where we've got to at this point now, the results are telling us that sex chromosome dosage effects on autosomal gene expression are not stable across cell types, but their effects on expression of sex chromosome genes appear to be largely tissue invariant. And I've already told you that the effect of sex chromosome dosage on expression of sex chromosome genes is primarily driven by these gametologs. So before we get into just the final imaging transcriptomic part of the talk, I just want to take a moment to think a bit more about these gametolog genes. These are the ones that have retained copies on the Y. So they have an ancestral copy on the X and on the Y. And these genes are interesting for a number of reasons, and this is some work from the lab of David Page. As I said, they're unusual in that they've been preserved across millions of years of evolution in multiple species. So for some reason, biology doesn't want to lose two copies of these genes, so it's hung onto them on the Y chromosome. When you think about the functional annotation of these genes, they all sit at key stages in the central dogma. They're key way stations in translating the genetic code to function. So chromatin modification, transcription, splicing, translation, and ubiquitination. And another interesting thing about these genes is when you look at the Y chromosome genes, some of them, the gametologs, are conspicuous for being expressed from the Y in multiple tissues, whereas all the other Y chromosome genes are testes specific. So these considerations taken together are telling us that you have this family of evolutionary preserved, functionally important sex chromosome genes that exist in one copy on the X, one copy on the Y. They don't recombine, but they have high sequence homology. They've been retained over evolution. They're broadly expressed across tissues. These are the genes within the sex chromosomes that show most consistent and stereotyped changes in their expression as a function of how many sex chromosomes you're carrying. And these changes in the expression of these genes seem to be relatively consistent across different cell types. And I think these factors make the gametologs particularly interesting for understanding how sex chromosomes could be impacting biological structure and function. So in the final slide, I just want to discuss this study that tried to jump levels of analysis and say, can what we know about sex chromosome dosage effects on gene expression inform what we see when we look at sex chromosome dosage effects on regional brain anatomy? And the specific way in which we're interested in that question is, why does this map look the way it looks? How can it be that you can change how many sex chromosomes a person is carrying? Some regions of the brain change their size and others don't seem to care at all. What accounts for those regional differences in brain vulnerability? And the hypothesis that we tested in this study, we call it the transcriptional vulnerability hypothesis. And it's been articulated by others too in other contexts. And put simply, the idea here is that could it be that those regions of the brain that seem to really care anatomically how many X chromosomes you carry, or they get really altered in patients with atypical numbers of sex chromosomes, that they have that vulnerability because in health, those X chromosome genes are particularly active in that brain region, for example. Therefore, when you mess with X chromosome count, they're the first regions to show troubles, if you like. It's a simplistic idea, but it's tractable. And it's plausible, given some of the findings from neurodegenerative disorders regarding regional vulnerability of the brain. So what this hypothesis would predict, if this hypothesis is true, is that those regions of the brain that show extreme anatomical change in patients with sex chromosome aneuploidy are those regions of the brain that show extreme expression levels of sex chromosome genes in health. And that's the hypothesis that we set out to test in this study that came out a couple of years ago, led by Jacob Seidlitz, who was a PhD student in the group then, now doing amazing work with Aaron Alexander Block in Penn. And Jacob, in this study, mashed together in vivo neuroimaging data with post-mortem maps of gene expression from the Allen Institute, which I'm sure many of you are familiar with. So the study design here is to take in vivo structural imaging data from patients with sex chromosome aneuploidy and make a map in the cortical sheet for how each cortical region is altered in patients compared to controls. Now, the specifics that I won't get into here is that this map is made using a technique called morphometric similarity mapping. But essentially, the thing to focus on for the purpose of the next few slides, we can discuss it more in detail if time allows, is that the extreme red regions show a prominent change in cortical anatomy in one direction, and the extreme blue regions are showing a prominent change in cortical anatomy in another direction when you increase sex chromosome dosage, and the white regions aren't really altered at all. So what Jacob did is he made these maps. He made one map for the way the brain changes when you add an X, an abnormal number of X chromosomes, and one map for the way the brain changes when you add an abnormal number of Y chromosomes. And then for each of these maps, he correlated them with the expression maps of 15,000 genes in the cortex as provided by the Allen Atlas. And he was able to rank these 15,000 genes for how similar their map is to this map. So at the top of the rank, you'll have a gene that is highly expressed in the red regions and low expressed in the blue regions. At the bottom of the rank list, you have a gene that's highly expressed in the blue regions and low expressed in the red. And in the middle of the rank list, you'll have genes that aren't correlated with this map at all. And now this enables us to test our hypothesis, because the test is, if we have this rank list of genes, when you generate the brain change map by adding an X, does the rank list of genes overrepresent X chromosome genes at its extreme? And we found that that was indeed the case. So here is the map of brain change when you add an X to humans. Here's the map of brain change when you add a Y. And this is showing the average, the median rank of the genes on each chromosome relative to this map. And the most extreme ranking chromosome for the X chromosome induced changes in brain anatomy were X-linked genes. And similarly, this extreme ranking chromosome relative to the brain change induced by adding a Y was Y-linked genes. So to put this in other words, what this seems to be saying is that the spatial patterning of cortical anatomy change in patients carrying abnormal numbers of sex chromosomes seems to be preferentially predicted by the spatial patterning of sex chromosome gene expression in health. And we thought that was a sort of intriguing result, and that didn't just hold for sex chromosome aneuploidies. In this paper, we look at 4 other types of gene dosage disorder and find the same phenomenon. Now, I've just told you in the early part of the talk that not all sex chromosome genes change their expression when you change X chromosome dosage. So if this model is true, if this transcriptional vulnerability model is true, there should be a prominent ability of dosage-sensitive sex chromosome genes to predict the anatomical change. So here we have the map of anatomical change when you add X, and we had identified in a tissue from patients with sex chromosome aneuploidy genes that change expression, and these red genes are prominently dosage-sensitive genes. And what we found is that the regional expression of these red dosage-sensitive genes as compared to the non-dosage-sensitive sex chromosome genes predicted the regional changes in brain anatomy. And then as a final test, we turn to inter-individual differences. So if it's the case that those brain regions that in health express sex chromosome genes that we know change their expression when you mess with sex chromosome dose, and that then when you mess with X chromosome dose, you do preferentially alter the anatomy of those regions. Could it be the case that if you take a group of patients who all carry an extra X chromosome, do those patients that show more severe expression change in their blood show more severe anatomical change in their brain? So to do that, we had a sort of unusual data set, quite a rare data set of 55 patients who carry an extra X chromosome, who had neuroimaging data, so we could score each patient for how severe their brain anatomy, how severely their brain anatomy is altered. And we also had measures of expression of dosage-sensitive sex chromosome genes in lymphoblastoid cell lines from these same patients. And we used PLS, partial least square regression, to model the relationship between the patterns of change in brain anatomy and the changes in gene expression peripherally. And that led to this summary plot. Each of these dots is a patient, is a participant. And essentially, those participants who had more severe expression change in their blood showed a more severe version of this expression change in their map. So working together then, these studies hopefully serve to at least suggest that there's some utility in linking transcriptional studies of sex chromosome dosage effects in cell types to macro scale studies of sex chromosome effects on brain anatomy, and that we may potentially, to some extent, be able to link together these two levels of analysis. So just to wrap up, I told you, I hope I've convinced some of you that and encourage some of you to follow up on this idea that sex chromosomes really open fascinating and empirically tractable entry points into multiple aspects of basic and clinical science. The sex chromosome aneuploidies offer rare, precious, naturally occurring models for accessing these windows in humans above and beyond the fact that they're medically important disorders in their own right. That when you study sex chromosome aneuploidies, you can show that they reveal that there's this family of sex chromosome genes, gametologs, that seem to be particularly sensitive to sex chromosome dosage across different cell types too, and which are worthy, I think, of prioritized study for driving sex chromosome dosage effects on phenotypes, either in the context of normative sex differences between XX and XY individuals, or in the context of sex chromosome dosage effects in aneuploidy. And that when we use these imaging transcriptomic approaches, there seems to be some evidence that if you know the sex chromosome genes that are highly dosage sensitive, they can help you predict regional brain changes in anatomy. And just some next steps to give you a feeling of where we're taking this next. We're trying to expand the sample size, the data modalities, and the cell types with which we're characterizing sex chromosome dosage effects on genome structure and function. We have a small number of post-mortem samples from brains of patients with sex chromosome aneuploidy, so we can directly study sex chromosome dosage effects on gene expression in patients in the brain. And I'll probably skip past these two, just to leave a little bit of time, as I'm afraid I ran a bit long. But I want to make sure I have time for the most important slide, which is to thank you all for your attention, and to thank everyone in the lab and collaborating labs who've done this work, and it's been a pleasure to work with and learn from. A huge thank you to the families in the study, without whom this work wouldn't have been possible, and a special thank you to my own family, without whom this work also wouldn't have been possible. So thank you to Shirley and Alma, and thanks to you. I'll take questions if there's time. Sorry if I ran long.
Wonderful, thank you. I see, David, you have your hand up, and you can start things off, and others can jump in afterward if you have questions. Feel free to speak up.
That was terrific. So I'm wondering whether, if you look at an individual who has normal number of X and Y chromosomes, but has autism spectrum disorder or something, a neuropsychiatric disorder of some kind, are the gametolog genes differentially expressed? Are they upregulated abnormally?
To my knowledge, gametologs aren't enriched amongst the genes that have been reported to be differentially expressed in neurodevelopmental disorders, say from some of the post-mortem work that's been done by the Geschwin lab, for example. But I have to confess, I haven't actually directly tested that. I don't know if you know, Donna, whether maybe sex chromosome genes in general, if they seem prominent.
Yeah, different between cases and controls. I don't remember seeing that reported, but could take a closer look at the data and see if that comes up.
Thanks for the suggestion.
Question.
I had a quick question.
Sure. Go for it.
Thanks. Which is, could the enrichment of gametologs be partly because Y gametologs cannot be X inactivated, and so if they share a function, then the X associated need to escape X inactivation to maintain sex expression parity?
Yes, so that's one of the ideas as to why, if you look at the, thanks for that question. It's a good one because it sort of highlights the fact that, which I didn't convey in the presentation, that when you think about the X chromosome and you classify X chromosome genes based on whether they escape X inactivation or not, those genes that prominently escape X inactivation and therefore are expressed from both X's are enriched for the gametologs. Almost all of the gametologs escape. Therefore, you have a situation where there's two copies expressed in males, the X member and the Y member, and two copies expressed in females, the two X's. Is that answer?
No, yeah, that makes sense. Thanks.
But then the interesting question becomes, although the X and the Y members, so you've got two X copies in here, and you've got an X and a Y, so you could say, well, we're good. They're equal in dosage. But the interesting thing is the X and the Y member are no longer recombining. They have high but not perfect sequence homology. But there's also the theoretical potential that they've undergone differential selective pressures for the way they're regulated, for example. And that's some work that we're doing right now in GTEC seems to suggest that actually they don't behave the same way, the X and the Y member. So that even though that they occur in two doses in males and females, the fact that males have an X and a Y member and females have two X's could be relevant for thinking about sex-biased biology.
Do they, could they be highly pleiotropic? Like, do they have potentially like shared function and then a differentiated function where the, like are there tissue types where they tend to maintain expression level parity and tissue types where they tend not to?
That's a great question. Yes, there are tissue specific effects in the level of their expression, but also how they're co-expressed. So the Godfrey paper would be a great place to look first. Godfrey, 2020, Godfrey and Page in Genome Research, I believe, where they look at levels of expression, but we've been sort of extending that, looking at co-expression. That's a great question. And some of the divergences are more prominent in tissues that show sex-biased organization. Thank you for those questions.
Thanks.
Hi, may I ask two questions? So the first one, I maybe missed this in your talk. I was late for a few minutes. I'm sorry about that. So my first question is, did you look at the number of like for the cells with extra X chromosomes, do you like how many barred bodies in those cells? Like, do they form extra barred bodies or?
That's a great question. Yes, I didn't include that for reasons of time, but Tom Mistelli, who works in the intramural program in the National Cancer Institute, so a kind of sibling institute to our National Institute of Mental Health, is interested in genome structure, particularly from the perspective of cancer. And they developed a technique for high throughput cellular imaging of chromosomal territories in nuclei. So we collaborated with Tom in a series of papers where they asked just that question. So we took fibroblast cell lines and basically tagged the X territory, the Y territory, and the 18 territory. Chromosome 18 is a control. And what that suggested is that there's in fibroblast cell lines, in relatively early passage fibroblast cell lines, X inactivation seems to behave pretty well. So if you look at these lines with multiple X's, everything other than one X is shut down, compressed, and peripherally located within the nucleus. There's an open question as to whether that behaves in model in vitro systems like iPSCs, and we had some interesting discussions on that topic earlier. Just a side note, one of the fascinating findings that came out of Tom's study, do you remember I showed you on the, I don't know if you were there for that bit, but there was this one family of genes that are annotated as being inactivated, which would paradoxically were more expressed from groups with one X chromosome than with groups with many X chromosomes. And this idea was that when you have the inactive X's in the nucleus, there's some trans effect that they somehow sort of suppress the expression even from the active X. And this was a transcriptional idea that came up from the expression data. What Tom's study showed is that the size of the active X territory was negatively related to how many inactive X's there are in the nucleus.
Wow.
So the size of the active X territory in an X monosomy line was larger than the active X territory in an XXXX line that had 3 inactive X's, and there was a dosage effect. However many more inactive X's you had, the more compressed the active X territory was. So there's this sort of, which is almost like a structural visual analog of the transcriptional results that I was presenting. So it does behave, but it seems to be messing with the remaining active X somehow.
That's so interesting.
Yeah, and I don't think anyone really, it's a total mystery. It's like what is going on? How is that kind of, what's underpinning that?
Thank you. And my second question is about, you mentioned about the volume of different brain areas, the change with the sex chromosome differences. Mm-hmm. So my lab study Rett syndrome, which is caused by a loss of a X-linked gene. And in Rett syndrome mouse, at least, they are have smaller brain size and they have reduced neuron size, reduced nuclei size. So I would just wonder, have you ever looked into your induced neurons to see if they're a cellular morphology change, things like that?
We haven't yet, but that's a great question. I mean, as you may have come across in the talk, we're kind of perhaps dangerously trying to span many different levels of biological analysis in the group. So an important kind of self-editing role is kind of figuring out where do we really have to draw the line and say this is territory we can't sort of wade into. Yeah. I think we feel reasonably comfortable trying to characterize these models transcriptionally, but we really don't have skills within the group for morphological analysis at the cellular level. But the hope is that we can donate these lines to banks to enable other groups to characterize all the many other phenotypes that these cellular models could provide.
I see. I think it would be interesting to see maybe there could be difference in neural subtypes. I think that would be interesting.
Yes.
Thank you.
Totally agree. Thank you.
Well, thank you all for your questions. We're 5 minutes past time, so I think I know the graduate class is going to stay on for a longer discussion, but for the rest of us, thank you all for attending. Thank you again, Armin, for your great presentation. And
sorry I ran a bit long.
we're all good. Yeah, so if we can give a virtual round of applause for Armin.
Thank you, everyone.
Thanks again for your visit.
Thanks.
We appreciate you
Some
being here.
great questions and for your attention. Thank you.
So this is pretty informal. We'll just wait for people to drop out, Armin.
Cool.
And students just ask the questions. I'm just a hitchhiker.
And I'm going to be one that has to drop off to go do daycare pickup, but enjoy.
Thanks,
Thank
Donna.
you, Donna. Thanks so much for the invite. It was nice to see you again.
You too. Take care, everybody.
Yeah, the questions were awesome. So one of there are many advantages to working in the intramural program, but one of the disadvantages is there isn't it's not a degree awarding institution. So there isn't a kind of large corpus of undergraduate and graduate students. We have the large student body intramurally is at the master's level. So people who've typically done their first degree come as called post-bac ERTAs for a couple of years. That's a very large and well-developed student body. And then we have a series of graduate program partnerships where students can come and do a PhD for 4 years, 2 years at the NIH and 2 years at another institution. And some of those are between the NIH and Oxford and Cambridge in the UK, or the NIH and UCL. But there are also schemes within the US. And then there's a large, rich postgraduate student body. But so I thought that might be interesting for the folks, sitting in, just to get a sense of what different institution environments are like. But I loved your questions, so I'm excited to talk more about them, or to answer any questions you might have about, the NIH as a training institute.
Okay, go ahead.
Hi, I have a question. Okay, so from what I understand, like the brain samples came from a UK brain bank. Is that right? Just before I ask my question.
Yeah, sure.
Okay.
So one of the studies that we published, we're recently starting to look at the UK biobank. So the brain studies for this paper that was led by Travis Mallard in Nature Neuroscience, looking at X chromosome dosage effects, X chromosome influences on the human brain, that was using the UK biobank. The studies that I was presenting in the talk just now focused on brain imaging data that came from rare patient groups that we fly into the NIH, and their patients that had abnormal numbers of sex chromosomes. So in a way, if you think about imaging data, these are two kind of complementary and distinct but related routes to saying, how does the X chromosome influence the brain? One strategy you can take is you can recruit patients that have abnormal numbers of X chromosomes and say, how does brain anatomy change as a function of how many X chromosomes you have, for example? And that's a kind of clinical genetics approach, relying on rare samples. Or you can take a statistical genetics approach using large data sets like the UK Biobank, and use these approaches like partition heritability to say, are there regions of the brain where the X seems to be punching above its weight relative to its size in terms of how much variance it predicts? Interestingly, those two maps aren't copies of each other.
Yeah, that's okay, because what I was gonna ask is like, okay, so if you're taking, for example, like these rare patient groups, I would wonder like under what conditions or like for what reason were they initially karyotyped? Because like that would mean that they were initially exhibiting some type of phenotype.
Great.
And then you might not capture people that weren't exhibiting whatever phenotype, and that would have a different expression, but might still be aneuploidy.
Yeah,
right?
Yeah, that's a brilliant question. Yeah, that's one of the problems we grapple with when we're studying these rare groups. So there's a couple of parts to the answer to that. So absolutely, if we put aside brain anatomy, if you take behavior, just like you say, if it could be that the only reason they know they have an aneuploidy is because they got tested, and the reason they got tested is because they had the clinical phenotype. So if you only phenotype people who know they have it, you're probably going to get a skewed view of the phenotypic range. One way and the only way of doing that is to, well, the optimal, the perfect solution to doing that is to try and ascertain everyone, even though they know, if they know they have aneuploidy or not. You can kind of get close to that using Scandinavian countries that have national registers. So we're collaborating with Denmark, which where they have this ISIC study where they can ascertain within a sampling frame every child with an aneuploidy, and then they can link that to whether they got diagnosed with a psychiatric condition or not. So there's no ascertainment bias in that study design. And even when you strip ascertainment bias out that way, you still find prominent associations. So there's a prominent risk for psychopathology, even when you've washed out ascertainment bias. What we do in our group to get a handle on ascertainment bias is we take all of the aneuploidy participants we can ascertain. Now, they all know they have an aneuploidy, but some of them got diagnosed with an aneuploidy in utero through prenatal testing. So the diagnosis wasn't as a consequence of a behavioral problem. And others got diagnosed postnatally, often because they had a developmental problem and they got tested. And we can compare those two groups to see, is there evidence of ascertainment bias? And we do find that the phenotypes do, on average, tend to be more severe in the ones who are ascertained because they had a problem and they went to. Having said that, there is still a significant phenotypic alteration in the ones who were diagnosed prenatally, and the profile of phenotypes is very similar between the two groups. With regards to the brain anatomy, I think we should actually do that stratification in the brain anatomy data. Groups that have studied rare genetic disorders to date and done the stratification find that there isn't really a brain anatomy effect. The brain anatomy doesn't really show differences in severity as a function of whether they were ascertained for behavioral problems or prenatally.
Okay. Along that line, I don't mean, sorry, anybody, but I'm just like really interested in this. But I was wondering, so for the brain maps of like the different sizes, I was wondering if those are also taking into account like at whatever age those brains were collected from the people and the different ways that their lifetime development might have changed the shape of the brain based on their experiences rather than necessarily like
the genotype.
You know,
Great question. Yeah. So because these are rare groups, the ideal study design would be to get loads of people and to scan them repeatedly over their development. And that's difficult when you're dealing with a rare disorder. So some of these conditions occur in like 1 in 1,000 births. So we have to make do with what we can. So the data are typically cross-sectional. So the individuals are aged between like 5 and 25. There are a couple of questions. So one question is the main effect of X chromosome dose that we observe, to what extent does it look like what we would have seen if we looked at 1-year-old? And the only aneuploidy that's been done for is Turner syndrome. And what that suggests, the neonatal work has been done by Rebecca Nickmeyer. And fascinatingly, Rebecca found in neonates with Turner syndrome a map of anatomical change that was almost identical to what we see at 20 years old. So at least with regards to that disorder, there seems to be quite a stable regional pattern, but we haven't had a chance to test that for the other aneuploidies. The other question you ask is to what extent could these changes reflect environmental effects? So for example, you could hypothesize you have an extra X chromosome. It gives you, let's say you have ADHD problems. That changes the way you're treated, the type of life you're able to, the type of environments you experience. And that is the proximal thing which changes your brain anatomy. One way we try to get at that, to examine brain anatomy in a way that washes out some of those signals, is because we have multiple aneuploidies, you can look at the effect of an extra X in different contexts. And those contexts don't all have the same behavioral consequences. So I can say, only show me those brain regions that change their anatomy when you, across several different contrasts that all change in X dose. So XX versus XXX, XY versus XXYY, XXY versus XXXXY. And that gives you a bit more confidence that we're really talking about the gene dosage effect here. And from what we can tell from those analytic study designs, these effects really are about gene dosage rather than some environmental mediation.
Okay, thank you. That's really interesting.
Great questions. How shall we, I'll just run through, I'm afraid I didn't note the order, so I'm just going to run through the screen as they come, if that's okay. Harshini is next.
Yeah, great talk. Thank you. I kind of had two questions. I guess I'll start with one and then ask the other one after you're done answering. So my understanding is that you're mainly a bioinformatics lab. So I was a little curious, have you thought about maybe like collaborating with someone who can like differentiate ESCs into either neurons or kind of like a more broadly neuronal directed path, using female and male ESCs alone, and just kind of like what you would expect to see there? Like, would you expect one to not differentiate as well or something like that?
Embryonic stem cells or
Yeah.
iPS? Yeah. Well, the embryonic stem cells obviously are rare within the sex chromosome aneuploidy participants. Right. The closest we come to that within the lab is that when we recruit the participants, they come to the NIH for 2 days, the participant with their family members, and we'll get detailed cognitive and behavioral measures. We'll get multimodal neuroimaging, but we'll also get blood and skin. So on a sample of them, we have skin tissue that we've reprogrammed to iPSCs.
Okay.
And we've started the process of differentiating those to neuronal progenitor cells. So we're only partway along that chain. So we now have transcriptional data at their LCL stage, lymphoblastoid cell line, transcriptional data and some methylation data in fibroblast stage. And we literally 2 weeks ago sent off the iPSC biosamples for RNA-seq, method-seq, and attack-seq. Okay. And we're currently having we're not doing it because we don't have that expertise in the group. We've outsourced it to a company that's generating. New York Stem Cell Foundation is driving the iPSCs to neuronal progenitor cells. Okay. So is that the type of study design you were?
Yeah. 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 I mean, I was talking more about mouse ESCs because I didn't know how available that would be, and I know that you mentioned the iPSC neurons in your talk, but I was just curious how available that would be with so many different aneuploidies. But yeah, that makes a lot of sense to do the reprogramming yourselves. It's a lot more accessible, I guess.
Yeah.
Yeah. And then I had a follow-up question because I noticed in a lot your gene lists with that are sex dosage dependent. I saw that Cadmium 6C and the Cadmium 5s came up quite a bit. And I work on Cadmium 3A and 3B. So, and I know that Cadmium 3A has a role in sex reversal, and Cadmium 3B SNPs have come up in GWAS studies for autism disorders. So I was kind of just curious to hear your thoughts on kind of the interplay between the epigenetic modifiers, sex chromosome dosage, and intellectual disability, kind of like all together.
Yeah. Well, the X chromosome is enriched for genes that can impart intellectual disability in loss of function mutations.
Okay.
I haven't tested whether that statement is also true when you restrict consideration to the gametologs. Or whether it's particularly true for X chromosome genes that are important chromatin, modifiers. So I think that would probably be if I'm understanding your question correctly, the way to kind of basically say, is there a particular capacity of these genes, either based on the fact that they're gametologs or they've been annotated to regulate chromatin conformation and gene expression. are those particularly enriched for their capacity to lead to neurodevelopmental disorders. In Travis's study, where we were using partition heritability analysis to kind of say, are there brain regions where the X-linked genes seem to punch above their weight? We were interested in saying, is that enriched capacity of the X to predict anatomical variation particularly prominent for gametologs? But there were too few of them to be statistically powered to do it. There's only 16 of them.
Gotcha, yeah.
There are some experimental models. So Art Arnold is a phenomenal figure in the world of biology of sex differences. And he's developed mice that alter in the numbers of sex chromosomes they have, but also mice that have individual knockouts of some of these key X-linked genes. So that might be another way of kind of getting at their biology. Does that answer the question?
Yeah, it does. Thank you so much.
Pleasure. Thanks for the questions. Elsa is next on the screen. Hey, Elsa.
Hi, thanks so much. Really enjoyed your talk. I also have two questions, which will hopefully not be too long. The first is, in general, were the brains within karyotypic usually like fairly consistent amongst themselves, or was there kind of a lot of variation even amongst people with the same chromosomal pattern? And the second was, I think one of the ones we submitted to you early, which is, do you think it's more accurate to say that there are a lot of disease-causing genes on the X chromosome, or that there are a lot of really important genes on the X chromosome, the misregulation or the abnormal regulation of which can cause a lot of serious consequences?
A great question. So let's take one at a time. So the question of variability. There is substantial anatomical variability within each of these aneuploidy groups, as there is within XX or XY, or self-identified male and female groups in non-aneuploidic populations. And there's some interesting work, I think, to be done that we've not yet done on, is variability itself modified by how many sex chromosomes you have? But nevertheless, there is substantial variability. So the maps that I show you have changed. That's about a difference in the mean. This question of variability within a population that has a gene dosage disorder is so important, because let's not think of the level of anatomy, let's think of the level of behavior. On average, having an extra X chromosome, for example, shifts your average IQ down by about 15 to 20 points, 15, depending on what control group you use. So there's some evidence at the group level that it's problematic for development, but there's huge range. So we, for example, saw 65 participants with an extra Y chromosome, and the IQ ranged from 50 to 120. The mean was shifted down, but the range was quite prominent. And from a family's perspective, from a medical perspective, what you want to know when your child is diagnosed with a sex chromosome aneuploidy is what's my child's IQ perhaps going to be, but is my child going to have difficulties or not? So the bar that sets for us is, and it's important, I think, to describe the mean effects of these genetic conditions, but we really want to get better at being able to predict where a particular person is going to end up, either with regards, perhaps more importantly, from a clinical perspective, their mental health rather than their brain size, although the brain size variability is interesting in itself. So we're doing a fair bit of work to try and predict that variation. And it turns out that if you, for example, with IQ, if you know the family background IQ, it helps you better predict where a particular person is going to end up. But before I move on to the next one, was there your question about variance? Was there a particular kind of framework within which you were asking that?
No, I was just curious. I was thinking about the question that Angel asked about kind of other environmental factors, and the brain just seems like something that would be influenced in so many different directions. I was curious. It would have been really, really, really surprising to me if you had said these were very, very consistent.
Yeah. So we have And then that opens up a very interesting set of questions about what might the sources of variability be in the context of a sex chromosome aneuploidy. You have background genetic variation, background environmental variation, but then maybe there are unique factors in the context of aneuploidy that explain why one person might be more severely affected than another. For example, are there individual differences in how good you are at shutting down X chromosomes? And could that be a unique source of variance in aneuploidy? So your second question was about, is it more accurate to say that the X chromosome is enriched for disease-causing genes versus genes that have important biological roles and when mutated can cause disease? Is that correct? Mm-hmm. That's an interesting question. I mean, I think to some extent that's a little bit about language usage and kind of shorthands that we use. So people will talk about autism genes. Well, there aren't really autism genes because there aren't any genes that only ever give you autism if you take them, but you never see intellectual disability or ADHD. So I think it's probably more accurate to say that the more complete and unbiased description is that these genes seem to be playing important roles. One evidence of that is when you mutate them, you can have extreme phenotypes in humans, such as intellectual disability. But the disease, the genes on the X chromosome that have been associated with intellectual disability when mutated, when you have loss of function mutations, often impart a whole number of other phenotypes. There can be dysmorphology, there can be a range of other endpoints. So I think I'd probably describe it that way, which suggests that they are developmentally important in different contexts. However, they all seem to share the property, these severe disease phenotypes linked to the X often share in common the property of intellectual disability, whereas they don't often share in common the phenotype of syndactyly or hypertelorism. So there seems to be something about the brain as an organ that can kind of concentrate the deleterious consequences of mutations in these genes. And that might reflect the fact that, you know, you could argue, well, isn't that just saying that these genes are biologically important and the brain is the canary in the mine? It's a particularly complicated organism that it's always going to go if you mess with an important biological process. And that could well be the case.
Thank you.
Thank you. Great questions. And Nathan is next on the screen.
So Nathan?
Yeah.
Yes. Do you mind waiting?
Sure. I can jump to Ceci. Is that in the class? Oh, no worries. Is Ceci, is that the correct pronunciation?
Yeah.
Hi, Ceci.
Yeah, thank you. Great talk. So I have a quick question. So why some genes are sensitive to sex chromosome dosage while others are not? Have you done any like gene ontology enrichment analysis and you found any enriched geoterms on the sensitive genes versus non-sensitive genes?
It's a great question. Thank you. So we can think about the X and Y separately, perhaps. Mm-hmm. Let's maybe start somewhere else. Let's start with the gametologs. So what I've told you so far is that from the data we have, the gametologs seem to be particularly dosage sensitive. So one might ask, what are the characteristics of these genes that could give them, be associated with this property? And because there are so few of them, traditional methods for Go analysis often won't reveal a statistically significant association because there's only 16 of these genes. But if you look, as the Page Lab had done, at the individual functional annotations of these genes, remember that kind of weird Venn diagram I showed on one slide? Mm-hmm. What's prominent is that these gametologs that arguably are the most dosage sensitive sex chromosome genes all seem to be involved in kind of key way stations in the transition from genetic code to protein. Now, that's a little bit hand wavy. That's not got a p-value associated with it. Mm-hmm. But the current thinking is that one reason that they might be particularly dosage sensitive is that they aren't subject to X inactivation because you need to have 2 copies. And that needing to have 2 copies represents the fact that they're very haplosensitive because they're involved in these core processes of chromatin conformation, gene expression, translation, etc., transcription, and post-translation modification, that you need to have 2 copies, and there's a real problem if you only have one of them expressed. Having just one Y chromosome isn't viable, for example. And that it is then the accompaniment of that is they're not inactivated, therefore they're dosage sensitive in groups that have abnormal numbers of sex chromosomes. Okay. So I think that rather than they're involved in a kind of high level downstream part of biological organization, like the synapse, for example, or it's more that they seem to be involved in these elemental processes. Does that answer the question?
Yeah, that makes sense. Thank you.
Hey, Emmanuel, you're next online.
Hi, great talk, by the way. Love the intro. Yeah, so your presentation really got me thinking about mosaicism and how this translates, like the differences in expression in this X and Y included genes. Like, how does that work for that type of genotype? And also, I was wondering, did you look at genes that were differentially expressed outside of the X and Y chromosome? And also, are these genes like similar to genes that control brain morphology?
Great questions, thank you. So the first one, mosaicism, when we recruit participants, we intentionally limit it to people who don't have mosaicism because it's kind of messy enough already. So we're kind of like trying to limit it to, but that's one of the to come back to the question that was asked earlier about phenotypic variability. Mosaicism is one possible mechanism of that. So that even though the participants that we have in their blood weren't mosaic, so we looked at 50 metaphase spreads and they all had to show the same karyotypic abnormality, the mosaicism could exist in other organs that we're not capturing. So it might be, for example, that one reason that a person with 3 X chromosomes is less impacted than another is the proportion of the cells in their body that have that karyotypic abnormality. So I think of mosaicism in the context of a potential mechanism for explaining variability in the clinical phenotype, but it's very hard to assess in humans because you can't systematically access multiple tissues. I guess you could if post-mortem. I can come back to talk more about mosaicism if you had other questions, but just in interest of time, just to go through the others you asked. The second one was, remind me the second one, sorry.
That's fine. If the genes outside of
yes,
the X and Y chromosomes,
yeah.
So
yes, I focused today on the local cis effects of sex chromosome dosage on sex chromosome expression, but many autosomal genes are dysregulated too. And that's been best characterized, I think, in the published microarray study, but will also be touched upon in the RNA-seq study that's coming out. So there are modules of functionally enriched co-expressed autosomal genes that seem to shift in their expression as a function of how many sex chromosomes you have. We've characterized those modules so far in lymphoblastoid cell lines. So that's not the ideal setting to understand whether those, or to what extent those autosomal genes might be relevant for some of the neuronal or brain phenotypes that we're seeing, for example. Hopefully we can address that in some of the iPSC-derived tissues that we have. Your third question was about the anatomical change.
Yeah, it was kind of related to the second question, but I was wondering if those genes, the autosomal genes that are differentially expressed, if they were also related to like brain morphology.
That's right. That's a great hypothesis to test. So those high-ranking genes that have an intrinsic expression in the brain in health that looks like the anatomical change map in patients, did those genes also include the autosomal genes that are differentially expressed?
Mm-hmm.
In fact, that's part of the question, but we part of the issue there is that, again, it gets into the fact that we've only been able to characterize autosomal effects in non-neuronal tissue. So I think that it's easier to concentrate on the sex chromosome effects because they seem to be tissue invariant.
Yeah, I would assume that would be a very hard hypothesis to test.
Yes, yes.
Well, thank you.
No worries. Angelica is next on the screen. Hey.
Hi. I was really interested in your work looking at kind of sex dosages on like structures of the brain. And I was curious if kind of some of the like brain regions you've identified, if they have known functional roles, like known functions relate to kind of like things like the disorders you see, or if they relate to known male versus female differences.
Thank you. That's a great question, Angelica. So the way we've looked at that question so far, because it was on our minds too, is we generated the map of regional anatomical change as a consequence of sex chromosome aneuploidy. And then we looked at the map and said, well, what do these regions do? Are they particularly involved in certain domains of function? So for example, if all the changes were only in the visual cortex, that might prompt the hypothesis that that would have a consequence for visual functioning, for example. And traditionally, the way that people address this question is they kind of reference their memory bank. They were like, oh, look, there's a anatomical change in this bit of the temporal cortex, and that's kind of been associated with language, so we predict that it could have a language effect. Recently, there have been these amazing tools that have been developed for doing that more systematically. I don't know if you've heard of it. Have you or anyone else on the call heard of neurosynth? The link in here. It's very, very cool. I can actually show you. So this is a website that allows you to do online meta-analysis of neuroimaging data. So the author of this tool went into the literature and scraped data from thousands, 10,000 plus neuroimaging studies, functional imaging studies. And they recorded which bits of the brain the paper reported changed activation. And then they scraped the text of the paper and they said, what terms are overrepresented in the text of the paper? So then you're able to say, if this brain region changes activity, what terms would tend to be overrepresented in the paper? Or you go the other way. If language is overrepresented in the paper, which brain regions show a change in activity? So for example, you could say, here's a picture of the brain. I can pick this part of the brain and I can say associations. And it says, these are the terms that are overrepresented in the paper if that bit of the brain is activated. So what we did is we used neurosynth to decode the anatomical change map in aneuploidy. We took the hotspots that showed convergent effects of sex chromosomes and we stuck them into the tool and we said what tasks tend to be associated with these regions. And we found that there was an overrepresentation of networks that are involved in affective regulation and language processing, mood and decision making was one set of regions. Language processing was the other. There were four, I'm trying to remember now. Those were an early study. I can't believe I've forgotten. It must be late in the day effect. There was decision making, there was language processing. I'm going to have to find out now. I'm sorry. I can't. So broadly speaking, the domains that we saw, at least superficially, were sort of relevant for the clinical domains that seem to be modified or particularly vulnerable in patients with sex chromosome aneuploidy. But that's a sort of distal level of evidence, right? You're basically saying the brain regions that are changed are known to be involved in these tasks. I'm afraid I don't have the map here. That's disappointing. I'm sorry about that. But the question you might want to push further on is to say, well, is it the case that if a patient has more severe alterations in this region, that area, then they have more, detection of biological motion? That's it. So this is a matrix. Each of these rows is a brain region that shows a convergent effect of sex chromosome dosage. Each of these columns is a word in the text of the paper. And the color is telling you that this word is associated with a change of activation in this region. So we clustered the matrix, and basically one set of regions in the temporal lobe seem to be involved in biological motion, another set involved in language, a cluster in autobiographical memory, and the reward affect interoception. So the domains, the brain regions that tend to be altered are not random. They seem to be involved in certain functional domains. But what we don't have evidence of yet, and we've looked, but the statistical signal really isn't that strong, is it the case that if you have a really big change in your language network, you have more severe consequences for impairments in language? And that may reflect a fundamental limitation in the ability, in the degree to which neuroanatomical variation in humans relates to behavioral variation. It may be that the measures we get from in vivo imaging just aren't good phenotypes for tracking behavior. We just have to take that on the chin.
That's really cool. Thank you for explaining all that. That's a cool tool too.
My pleasure. Yeah, you can play with it. It's like amazing. You can lose hours in the thing because you can pick a task and it tells you the network associated with the task, or you can pick a region and it tells you the task. It's great. So
That's
C1.
cool.
Oh, it is nice. Thanks for the question. C1 is next.
Thanks for the great talk. I have a quick question about, is the comparison between the specific distribution of gene expression and the distribution in anatomic brain changes along different aneuploidies informative of functional mechanisms of these genes? I'm specifically talking about genes that you've detected that have highly correlated expression with anatomic changes in severities in response to aneuploidies.
Yeah, thank you. That's a great question. So in the Nature Communications paper that described this work, we were interested in that question. So what we did was we take a brain map, we rank the brain expressed genes relative to the map, and then we can do go enrichment at either end of that rank list to say, are there particular terms or cell types that are enriched in these extreme ranking lists? And that did seem to, that was useful in as far as it prioritized some candidate substrates that might be particularly relevant. So for some rank lists, synaptic terms were highly represented. For other rank lists, axonal connectivity was highly represented. So we've generated those annotations, but we've not really dug into them much more than the initial active description. What we're particularly interested in along that sort of playbook, though, is the cell type approach. Because when we're trying to get at this problem through the in vitro models, we can make neuronal progenitor cells, but then there's a whole bunch of cell types in the brain that we could make. And the question is, and each one is like, a huge amount of money and time, which one should we make? And that's one reason, one place that we turn to annotating the correlated genes in the brain, because these different aneuploidy maps show differential enrichment for particular cell types. And they not only implicate a cell type, but we can then say, for this aneuploidy map, in the brain region that shows an extreme red change, this cell type is enriched, and a marker gene of this cell type that lies on the X chromosome is this gene. So even if you want to push it a bit further, gives you a targeted hypothesis of a particular gene within the X in a particular cell type that might be relevant in a particular region. So that's the most specified hypothesis the study design generates. Does that sort of align with the direction of your question? Is that touching where you were going, or?
Yes, actually, this answers the first half of the question. A follow-up one to this is like, let's say if we have targeted a gene with the method I just mentioned, can like the changes of the gene expression with the changes of the analytic brain changes give you further information about like if they increase if the genes increase as the anti brain increase, or the sort of distribution, would that be informative of how this gene works?
I mean, if the gene is highly expressed in a region that expands versus a region that contracts.
Mm-hmm.
Theoretically, yes. I mean, one caveat that I should make sure I don't forget to mention is, and it's interesting. It poses a problem, but it also, in somehow, it's a surprising alignment.
Yeah.
These aneuploidies presumably modify brain development way back early on in development. It's a reasonable guess to make that when an X is impacting regional development, it's doing it, back in prenatal time onwards. These transcriptional alignments that I mentioned in the presentation are based on gene expression in the brains of people 40 and 50 years old. So in some ways, that it's another reason that you would really not expect to find this alignment. So the fact that we do, even though we're looking at expression data in the wrong developmental period, is striking. And two possible interpretations of that observation are: one, the profile of expression that you see in 40, 50-year-olds is sufficiently similar to the spatial pattern of expression early on that you're still seeing the correlation. Or two, the fact that they're correlated in adulthood is telling you that there's still an active relevance of the gene dosage for the maintenance of abnormal anatomy. It's not like that it's etched in the brain and then it just plays out during development. So I think those two hypotheses have different implications for what you would predict and what you would want to investigate regarding the way in which a particular gene is driving the anatomical change. Part of the problem is you can't specifically manipulate individual, not the problem. I mean, the reality is you can't easily manipulate individual genes in humans. And that's why we accompany this human work with animal work collaboratively. So we have colleagues that do murine neuroimaging, and there are murine models where you can target specific genes. So that's a kind of a intact organ way of functionally annotating the genes implicated by the imaging transcriptomic methods we use in humans.
Well, that's very thorough. Thanks.
Pleasure. Thank you for the question. And that leaves Nathan.
Hi, can you hear me?
Yeah.
Great. So it sounds like from the context of your talk, you were talking about aneuploids that result from some mistake during meiosis where they've been aneuploidy through their entire lifespan. I know, for example, Y chromosome loss is an incredibly common somatic mutation in blood cells. Do you know of any examples where you found a somatic aneuploid mutation in the human brain?
Yeah, that's such a great question. No, not that we looked, but you're right. I mean, there's an interesting question. Well, the first thing just to say is that the aneuploidies that we study are believed to be de novo. They're very rarely inherited, and they're thought to be caused by errors in chromosomal segregation during meiosis in some individuals, but also in another proportion of individuals, and the percentages aren't easy to resolve. A segregation, a mitotic segregation error in the zygote in the early, very early development. So but nevertheless, that happens very early on. But you're talking about, so what that means is that depending on how early it happened, you're right. You could get a situation actually where the aneuploidy develops clonally in a sub portion of cells at some stage during postnatal development. And in those patients that are defined as being aneuploidic based on the fact that they have an aneuploidy in their blood or in an accessible tissue, what is really happening is in their brain, only some of the cells are aneuploidic and others aren't. And to assess whether that could possibly be the case, you'd need to have access to brains of participants with aneuploidy and have multi-regional sampling of the brains. And those data don't exist, but it would be a very interesting thing to look at. It's a bit of a black swan situation. If you just found one patient with an aneuploidy that had that sort of mosaicism, it tells you that it can exist. And I think the prospects for that type of developmental clonal mosaicism really depend on how late in the game you can develop a sub-line of cells with an aneuploidy. The other question about mosaicism in the brain, another possible way of getting at it is it could actually not be about regions, but it could be within a region. So we have a few individuals that we've identified in brain banks that have aneuploidy, and we've got single nucleus RNA-seq on those individuals in brain from dorsolateral prefrontal cortex and the lateral temporal lobe. So it may well be that when we look at those data, actually only some of those cells have the aneuploidy. And then the other facet of your question is this very interesting one about loss of a Y chromosome, particularly, and that I know is a progressive thing that happens in older age cohorts, for example, and that there's some phenotypic correlations of Y chromosome loss in the blood. So I don't know about that. I mean, I guess in a largely post-mitotic organ like the brain, the mechanisms for generation and maintenance of Y chromosome loss are probably different to what they'd be in blood.
Right.
But I guess it's possible that you could have somehow Y chromosome degradation or something in a post-mitotic cell. But these are fascinating questions that you're raising, Nathan. I think that what they call for is greater access to post-mortem tissue, brain tissue in patients with aneuploidy.
So a bit of a speculative follow-up question. If this kind of a mosaic of chromosome numbers within the brain is technically, I guess, it's not known whether it exists or not, but it's certainly possible. Like you had mentioned earlier, there's kind of this ascertainment bias where the people who are getting karyotyped are the people who come into the doctor with symptoms. Could it be possible that Y chromosome loss later in life does not manifest these symptoms associated with aneuploidy that is present through the entire developmental cycle?
Could be, yeah, you could certainly imagine how an abnormal gene dosage when you're building, the organ has a different consequence to an abnormal gene dosage down the line. It's entirely plausible. There are some data from Chris Walsh's group. That they're one of the groups that are working hardest to describe mosaic gene dosage variation in typically developing brains. I think they've also got some brains from participants with psychiatric disorders. I'll look more closely. Your question is prompting me to look more closely at their work to see whether they actually found any evidence of Y chromosome dosage variation. Let me try and I'll put the paper in the chat for everyone.
Yeah, that'd be fantastic. Thank you.
This is the biggie. Let's see if they actually mention sex chromosomes. It's a very good question. On a text search, but anyhow, yeah, that paper and related ones would be a good place to start looking.
Great, thank you for that recommendation.
Oh, did I put it in the chat?
You put a Nature paper in the chat.
Okay, yeah. And Liz is next.
Hi. Yeah,
Hi.
thank you so much for your talk. I really enjoy it. My question is mostly career-wise. How was the transition from being a doctor and a PhD student into going into postdoctoral program in the NIMH?
Thanks for asking that. Yeah, it's a these shared experiences of career development can be, I found them hugely valuable, you know, hearing from other people, and we all pursue such different paths through our training.
Yeah, I never heard about the intramural research program.
Yeah.
Why?
So so I trained in the UK. I got my medical degree and my PhD in the UK, and the MD PhD track in the US very often is done with the PhD being done early. So people will either do a PhD, then do medicine, or they'll do the first 2 years of medical school, preclinical, then break, do their PhD, then go back and do the clinical rotations like that. Whereas in contrast, it may be different in the UK now, but when I was doing it, people do the PhD later. I didn't do any science really until I was 30. So I did my medical training. I trained as a pediatrician, working on the front line, seeing kids, doing healthy baby checks, then retrained in general psychiatry. And then when I specialized in child psychiatry, you could do a hybrid fellowship, a hybrid child psychiatry fellowship, where half of the time you're specializing and you're seeing children clinically, and half of the time you're doing your PhD. So normally the child psychiatry fellowship takes 3 years, but if you do a PhD, it kind of takes 4 or 5. And that's really when I first started to do science in any meaningful way. And I didn't actually code until 2000 and properly code until like 2008, when I came to the NIH for the first time. So I would have been like 34. And there were some very patient folks in the lab who kind of helped me learn how to use R, and I completely, fell in love with it. But I'm mentioning that to say that it's interesting to reflect on how the way you combine clinical and science training often feels different depending on when you do that combination. So people in the US model, I've heard some people say one of the disadvantages is you can do your PhD in retinal disease, and then by the time you finish your clinical training, you're like, oh, I really want to be a hepatologist. So it's like kind of trying to link your PhD to your clinical work. Whereas when you do it in the UK model approach, you're really doing it, the reason I did a PhD was because I was seeing all these patients and just utterly fascinated by what I was seeing. I just wanted to go and know why. So you're really doing a PhD with intent in a way that is supported by some clinical knowledge, and you can fold it back into the clinical work. So that's an important thing to reflect on. And then the part of the question relates to the intramural program. It can sometimes be something that doesn't have high visibility in the extramural world. And I have to say, it's always a bit odd to me that that's the case, because in some ways it's a scientist's fantasy land. I think of the NIH like it's like NASA for the brain, essentially. You kind of have this stable, long-term, high-risk funding that enables you to do types of work that will be very hard to get funded. And this came up in a conversation with one of the faculty members earlier. The intramural environment enables you to do very interesting science, and you were asked to do science that can't be done under a grant funding mechanism, because otherwise they're not using the portfolio to its advantage. The advantage of that is that you get to follow the science. You can have a question and you don't need to wait to get funding acceptance for that particular project, or frankly, convince a funding body that you have any ability to do the work, which that could be a bit of a problem. So we get funded in 4-year blocks, and then we're assessed every 4 years. If it goes well, you get another 4 years of funding. But in that 4 years, I could, within reason, if I wanted to, pick some completely harebrained idea and follow it. Now, it's a problem if it doesn't pay off, but that's really exciting because you get to, it gives you a space where you can be quite creative and adventurous in a way that is harder when you have to justify the work in kind of little chunks, although you can do it. The downside of that model is that unlike the extramural world, you're not constantly having to really think deeply and justify every single step of what you're doing. So there's not that sort of sanitizing and effect of the rigor of grant writing. That's one downside. Depending on the scientist and the setting, I think for some people that can be a problem because you can kind of go off on strange tangents that really don't make sense, but no one's ever kind of forced you to. I think one way to counterbalance that is to continue. There are ways of continuing to apply for grants intramurally, even though you don't need to. And that's something that I do. I still write grants because I think it can really help you kind of focus your thinking. I think the other risk of the intramural model is that it can lead to situations where investigators can be investing a substantial amount of money in a technique that they don't really have a track record to deliver. And I think I alluded to that in the talk. That's something that I constantly have to edit or reflect on myself, because as you would have seen, in one study, we're doing imaging, and another study, we're doing transcriptomic and iPSCs, and they're like wildly different types of work. So I think the way to operate safely in that context is to be self-critical and be self-reflective, demand of yourself that you can show that you can deliver before you invest any more money in a particular method. If it's something new, convince yourself you can do it before you do it again. And also to build really rich collaborative networks. And I have to say, I think, not to evangelize, but I think for the right type of investigator who wants to do the right type of science, or as a trainee, the intramural program can really be an absolutely phenomenal place to work. From a trainee's perspective, you're not, they're fully funded. You don't have to get your own funds. You can have very stable funding for 4 or 5 years. And typically you're working with these jaw-dropping data sets that are jaw-dropping because they've been gathered in these very unusual funding contexts that lets you get really ultra-longitudinal data or ultra-deep phenotypic data on multiple rare groups. So it can be a very, very special place to train. What it doesn't have is the familiarity with applying for grants. It doesn't have an embedded undergraduate, and it's not a degree awarding institution. So in those ways, it's culturally a bit different from a traditional university setting. And for some trainees, that's been a concern, because the worry is if you come intramural, you can't go back out again and train extramurally easily. In the old days, that used to be a problem. But there are now funding mechanisms that enable people to jump into the intramural program, get the special training experience that is specially provided there, and still be able to be competitive when you transition out.
Okay, thank you so much.
Do you have energy to answer 2 short questions? I have.
Oh, yeah.
So the first question is, and I should know this, has anybody ever tried to do a forward genetics mutagenesis to isolate aneuploids? Even though sex determination is different in each different organism, but just to force to see what happens, how you can get aneuploid.
I see. You mean like a model organism?
Yeah.
That's a great question. Not that I'm aware of, but I haven't looked at that literature. very closely, but it would be I mean, I'd assume that there'd be the pathways involved in chromosomal segregation would kind of pop up quite high, but that's a great question. No, I'm not aware of it.
Right. Well, I was thinking more in terms of not the mechanics, I guess. I mean, obviously that's one mechanism, but sort of the thing you were talking about, which is, this happens early and
the clonal
compensation.
survivability.
Yeah. Because that might be one way to get at that.
Yeah, that's a very interesting question, actually. You could kind of find phenotype modifying loci in the context of aneuploidy. Yeah, it's a great idea.
The other question is totally about career development. So given all that you know, let's assume that you are most interested. I know you're interested in all these three different angles, but let's say you're most interested in sort of the gene disease connection, okay, whether it's gene dosage or whether it's just autosomal genes, doesn't matter. What would you advise to the current students as to things they might want to arm themselves with, tools or skills, or knowing all that you know?
Yeah. Yeah, thanks for asking that question. It's a provocative one. I mean, I think I'd probably say, given the way things are going, I'd say a deep training in computational biology, being comfortable with manipulating large data sets, version control, implementing statistical routines, working on clusters. I think those are, the skill sets that are going to enable people to then, once you have that arsenal, then as the biology speaks to you and you find the questions of interest, you can then deploy those skills on the many vast data sets that are growing. I mean, the UK Biobank, there's lifetimes of questions that are available in that data set alone. So I think that's the first thing I'd say. But then I'd also highlight, I think, the value of pairing that brute force bioinformatic capacity with I think it can work particularly well if paired with a biologically motivating question, is to still find a problem that really speaks to you in and of itself. Find a hard but tractable problem. Find a space that is the gap that's defined by work that's being done, and then bring those two things together. I think that's a really rewarding position to be in. I don't know if that answers the question, but that would be if I was talking to myself in the past, I sure as hell would have done it, and I would have done more math. I'd have done more math and I would have done more coding earlier on, rather than try to come to it when my brain is as addled as it is now.
Okay, well, any other questions? Those are really good. That was a really good discussion. I think everybody really enjoyed that. So once again, thanks so much.
Enjoyed it.
Thank you so much. And
thank you. And thank you all for your time. Those were just awesome questions. They really were.
Yeah, we have good students. That's
you say that again. Yeah.
Thank you.
Okay. Bye everyone.
Bye.
Take care.
Thank you.
Bye-bye.
Thank you.
Yeah.